Welcome back, students. In this section, I'll be talking to you about cholesterol metabolism. Nowadays, every lay person also knows about cholesterol. He will come and ask you what to do with cholesterol. So one thing is, for that sake at least, you should know about cholesterol metabolism. And the other thing is, it is important exam-wise. Thirdly, it is important clinically. So because of all these reasons, you will have to know a bit about cholesterol metabolism. So what is it? What is cholesterol? Cholesterol is a steroid nucleus. It is an animal steroid. We say that it is an animal steroid because it is having no plant sources are present for cholesterol. It contains cyclopentanoperhydrophenanthrene ring system. It's a big name. So what does it contain? It contains 27 carbon atoms. That's important. What is another feature of cholesterol? It is an amphipathic liquid. Is it synthesized in our body? Yes. By which cells? Only the liver? No, it is synthesized by all the cells within the body. Why? All the cells have cell membrane, and cell membrane contains cholesterol. And so cholesterol is an essential component of the cell membrane. Hence all nucleated cells synthesize cholesterol within the body. What is its role in the cell membrane? It is responsible for the fluidity and stability. One place we say stability, at the other place we say fluidity of the cell membrane. Depending upon the cholesterol content, the hydrophobic core will change. So it is responsible for both the fluidity and stability of the cell membranes. It helps to insulate nerve fibers. Hence, cholesterol is required. It is a precursor of steroid hormones. All the steroid hormones, whether it be estrogen, progesterone, testosterone, any of the steroid hormones requires cholesterol as its precursor. It is a precursor of bile acids. Bile acids are required for the digestion of lipids, for the absorption of lipids. Bile acids are required. It is a precursor of vitamin D, and vitamin D has got so many important functions in the body, including maintaining calcium homeostasis within the body. So for all this reason, cholesterol is an important compound which is required by the body. Now, one important feature I would like to point out here is that cholesterol is amphipathic, but cholesterol ester is not amphipathic. It is hydrophobic. Why? When cholesterol ester, it is cholesterol with a fatty acid esterified at the third position. Now we go on to the role of liver in all this cholesterol metabolism. Liver has a very central role. And what are the sources of hepatic cholesterol? By now you should be able to tell how liver gets its cholesterol. So which are the sources? Yes, one LDL is definitely a source. De novo synthesis of cholesterol is definitely another source of cholesterol. Whatever cholesterol that has come from the diet, how does it reach the liver? It reaches via the chylomicron remnants. So the different sources, dietary cholesterol comes from chylomicron remnants. Cholesterol from extrahepatic tissues comes via the reverse cholesterol transport via HDL. And de novo synthesis, which is taking place within the hepatic cells itself. What is the fate of hepatic cholesterol? What happens in the liver to this cholesterol? It may be transported outside to the extrahepatic tissues. How? By VLDL, LDIDL, and then LDL. LDL is the one which is transporting cholesterol to extrahepatic tissues. It may be directly excreted into the bile. So in the bile, please remember that gallstones are commonly precipitates of cholesterol. They occur when bile becomes supersaturated with cholesterol. So it is the ratio between the bile acids and the cholesterol which determines the liquidity or the formation of gallstones within the gallbladder. So usually this gallstones can occur when there is obesity, biliary stasis, infections, etc. These are only precipitates of cholesterol, most common. So bile acid synthesis and excretion into bile. So cholesterol is not only directly excreted, it can be also converted into bile acids and then excreted into the bile. So here I have given you a schematic diagram. We have the liver cholesterol pool which has got from diet, from de novo synthesis, and cholesterol synthesized in extrahepatic tissues, and this has come through HDL, and it is secreted out of the liver by the secretion of HDL and VLDL or LDL. It is goes as a free cholesterol in the bile, or it is converted into bile salts or bile acids and then excreted into the bile. Now, focus of this session is on the de novo synthesis of cholesterol. What do you think are required? From where does cholesterol synthesize? What are the prerequisites? What is required? Just as fatty acids are synthesized from acetyl-CoA, cholesterol is synthesized from acetyl-CoA. 27 carbon atoms is got from adding two molecules at a time. That is a 2-molecular acetyl-CoA. That is the importance of de novo synthesis. The steps are very long and complicated, just as cholesterol metabolism. So I'll just be giving you an overview of the cholesterol metabolism. Please remember, the major site for de novo synthesis is the liver, the intestine, adrenal cortex, testis, ovary. Why? In all these tissues there is synthesis of steroid hormones. So whenever there is steroid hormones to be synthesized by the testis or by the ovary, they need to produce more cholesterol, because cholesterol can be converted into these steroid hormones. Adrenal cortex, intestine, the same role. Basically, de novo synthesis occurs in all the nucleated cells. So which are the enucleated? Non-nucleated are the mature erythrocytes. So other than the mature erythrocytes, de novo synthesis occurs in all the tissues. It is mainly happening in the cytoplasm and partly in the endoplasmic reticulum. Now all the 27 carbon atoms are derived from acetyl-CoA. Approximately 1 gram per day is synthesized. Approximately 1 gram of cholesterol is synthesized per day. Now there are 5 stages in its synthesis. I am just going to give you an overview. Basically, again in MCQ point of view, how many acetyl-CoAs are required to form 1 molecule of cholesterol? 18 molecules are required. So 18 acetyl-CoA, we require energy? Yes, definitely. Do we require NADPH? Yes, definitely. Plus oxygen will result in the formation of cholesterol. Carbon dioxide, NADP, ADP, PI, all these are formed. So basically acetyl-CoA, energy, reducing bioequivalent. So all these will help in the formation of 1 molecule of cholesterol. Now, there are different stages, out of which one stage I am going to explain briefly to you, that is the synthesis of HMG-CoA. Till the synthesis of HMG-CoA, there is some slight similarity with another synthetic pathway, that is with the ketone bodies. Hence, I am going to tell briefly about the synthesis of HMG-CoA. Please remember, this HMG-CoA is a 6-carbon compound. This occurs in the cytosol. Please remember, this is a different HMG-CoA pool when compared to that of the ketone bodies. There is a HMG-CoA pool which is happening for ketone bodies present in the mitochondria, and the one in the cytosol is for cholesterol synthesis. Now basically the steps are when 2 molecules of acetyl-CoA, so remember they are formed from acetyl-CoA. So acetyl-CoA plus acetyl-CoA, 2 molecules. What happens? It forms aceto-acetyl-CoA. Aceto-acetyl-CoA. Aceto-acetyl-CoA reacts with 1 more molecule of acetyl-CoA. So there are 3 molecules of acetyl-CoA. 1 more molecule of aceto-CoA to form HMG-CoA. So these are the steps which are involved in the synthesis of hydroxymethyl-butyryl-CoA, HMG-CoA. Let us stick to the fact HMG-CoA. Now there are 2 isoenzymes of HMG-CoA synthase also. This step requires HMG-CoA synthase, and there are 2 enzymes of this HMG-CoA. So which are different. One is in the mitochondria and the other is in the cytosol. The one in the mitochondria goes for ketone bodies and the one in the cytosol is for cholesterol synthesis. Next, what happens? Now the HMG-CoA again is converted into mevalonate. Please remember, mevalonate is different from malonyl-CoA. Malonyl-CoA is something which is happening when acetyl-CoA is carboxylated during de novo synthesis. Malonyl-CoA is formed, which is an inhibitor of beta oxidation. Carnitine acyl transferase 1, which is an inhibitor of CAT1, carnitine acyl transferase 1. But now here HMG-CoA is converted into mevalonate. Mevalonate is done. This is done by the enzyme HMG-CoA reductase. This enzyme is a very important enzyme. Why? we have pharmacological agents which can inhibit this enzyme. This enzyme catalyzes the regulatory step of de novo synthesis. This is the rate limiting enzyme synthesis of mevalonate from HMG-CoA by using the enzyme HMG-CoA reductase. Now the clinical significance, as I said, there are drugs which are called as statins. One of the commonest is levostatin, which inhibit this enzyme, and they are given to decrease the serum cholesterol levels. So with this, I'll go on further. Till here is what you needed to know. What else do you need to know is that all carbons come from acetyl-CoA. 18 acetyl-CoA are required. It requires NADPH, ATP and oxygen. And the stages: first stage forms HMG-CoA, second stage HMG-CoA reductase to form mevalonate. In the third stage, it results in the formation of 5 carbon intermediates called as isoprenoids. Now in the fourth stage, 6 isoprenoids combine to form squalene. In the fifth stage, squalene undergoes several modifications. Squalene is a 30 carbon compound. It undergoes several modifications along with oxygen. There is an important enzyme, squalene epoxide, which helps in the formation of cholesterol. So this is what is needed to be known, because even other enzymes, none of them are there. There are a lot of molecular rearrangement, lot of NADPH is required during all these stages. That is the importance of knowing the cholesterol synthesis. One important thing is, how is this regulated? How is cholesterol synthesis regulated? Now, as I said, HMG-CoA reductase is a rate limiting enzyme. So which are the different factors which regulate the enzyme? Always we have covalent modification, we have induction or repression, or we have substrate availability. So in these spaces, and plus the feedback mechanism. So in this enzyme is regulated either by feedback mechanism, hormonal and nutritional. So I'll talk about all these things. So the different ways by which this enzyme is regulated is by induction or repression, covalent, modification. Different factors are these: feedback, hormonal, nutritional, and induction, repression, and covalent modification. So first, to talk about feedback regulation of cholesterol de novo synthesis. How is it done? So cholesterol itself inhibits its own synthesis. That is the feedback regulation. So after this, cholesterol is formed. HMG-CoA reductase forms. This enzyme reductase is inhibited by cholesterol itself. Thereby cholesterol regulates its own levels within the cell. So cholesterol is the feedback regulation. Mevalonate, which is formed from, is a product. HMG-CoA reductase converts HMG-CoA to mevalonate. Mevalonate itself inhibits the HMG-CoA reductase. Both of these, not only they inhibit it, they repress the synthesis of HMG-CoA reductase. They even go at the genetic level. Cholesterol is actually supposed to go at the DNA level and decrease or repress the amount of HMG-CoA reductase. Now, hormonal regulation. Cholesterol synthesis is increased by insulin. Minute you say increased by insulin, it means this enzyme has to be active in the dephosphorylated form. So it dephosphorylates the enzyme and makes it active. So you have to just know the pathway, and you have to know whether insulin is going to act on that pathway. Yes, whenever it is a well-fed state, everything else is also happy and they also are synthesizing. So in a well-fed state, insulin is more, cholesterol synthesis is more, and this is increased by insulin by dephosphorylation. The other opposite of that is glucagon. Glucagon acts via cyclic AMP, causes phosphorylation and inactivates the enzyme and decreases cholesterol synthesis. Are there any other enzymes? Yes. Are there any other hormones? Yes, thyroxine. Thyroxine causes induction of the enzyme. So that is why in hypothyroidism there is chances of getting hypercholesterolemia. So in hypothyroxine is needed to causes the induction of the enzyme. Glucocorticoids cause repression. Now, what are the nutritional factors which are responsible, which regulate cholesterol synthesis? It is the dietary cholesterol suppresses the synthesis of the enzyme. Increased caloric intake stimulates cholesterol synthesis by increasing the availability of the substrates, acetyl-CoA and NADPH. So with this, there is how cholesterol synthesis is regulated. Now we go further, how cholesterol is degraded. The most important portion, the degradation of cholesterol. Now please remember, degradation of cholesterol does not produce energy, and moreover the sterol ring cannot be broken. Once it is formed, the sterol nucleus is formed, it is not broken, it has to be excreted as such. So it does not produce energy and the sterol ring cannot be broken. So will it produce acetyl-CoA? No, it will not produce acetyl-CoA. So average diet has around 300 milligrams of cholesterol per day. Now around 700 milligram is synthesized per day. So totally we have around 1000 milligram. Out of this, 500 milligrams of cholesterol as such. Remember, cholesterol as such is excreted into the bile, and around 250 to 300 milligram is converted into bile acids and then excreted into the bile. So how is cholesterol excretion into the bile happening? As I said, around 500 milligrams per day. Please remember, the excess of cholesterol can form cholesterol stones, or gallbladder stones can be formed. Now this, whatever cholesterol is excreted, it is partly reabsorbed from the intestine. What happens to the unabsorbed cholesterol? Now the unabsorbed cholesterol is acted upon by intestinal bacteria to form cholestanol and coprostanol. These are called as the fecal sterols. The cholestanol and coprostanol. This can be an MCQ. Please remember that the unabsorbed cholesterol is acted upon by intestinal bacteria. So what is the clinical importance of this? As I said, vegetable oils contain plant sterols which prevent the reabsorption of cholesterol. See, remember, it is partly reabsorbed in the intestine, whereas vegetable oils contain the plant sterols which prevent this reabsorption. And also remember that excess cholesterol in bile can lead to the formation of gallstones. Now we go on. One is cholesterol is excreted as cholesterol. The second mechanism is cholesterol is excreted by the formation of bile acid and bile salt. So a bit about the bile acid and salt metabolism. They are synthesized in the liver from cholesterol. Now these are the 24 carbon atoms and they are the excretory forms of cholesterol. Now bile acids or salts are involved in dietary lipid digestion as emulsifiers. Now there are primary bile acids, primary bile salts. Let me tell you about this, and secondary bile acids and secondary bile salts. Let me clarify this. Primary bile acids are cholic acid and chenodeoxycholic acids. And the secondary bile acids, look at that, are deoxycholic acid. You remove that cheno, it becomes deoxycholic acid is converted into deoxycholic acid. And chenodeoxycholic acid is converted into lithocholic acid. Now, what are the primary bile salts? Primary bile acids, we saw that it is cholic and chenodeoxycholic acid. What are the primary bile salts? Primary bile acids, when they are conjugated with glycine or taurine, like it may be glycocholic acid, taurocholic acid, glycochenodeoxycholic acid, taurochenodeoxycholic acid are the primary bile salts. What are primary bile salts? Primary bile acids conjugated with glycine or taurine are primary bile salts. What are secondary bile salts? Secondary bile acids conjugated with glycine or taurine. So they include glycodeoxycholic acid, taurodeoxycholic acid, glycolithocholic acid or taurolithocholic acid. So how do you remember them? Please remember CC, that is cholic acid, and chenodeoxycholic acid, whereas others are all you need. For the cholic acid you need some other prefixes, like for secondary you need deoxy or it may be litho. Always remember cholic acid is a common component. Cholic acid, cholic acid, cholic acid, cholic acid. So cholic acid, cheno means it becomes primary. Litho or deoxy, it becomes secondary. And with this, when they are conjugated with glycine or taurine, they become bile salts. Primary bile salts and the secondary bile salts. So bile acids, please remember, they are amphipathic liquids. Now how are they synthesized? Cholesterol is hydroxylated at the 7th position by 7-alpha hydroxylase. This is the rate limiting enzyme. Again I tell you need not know the pathway. You need not know how cholesterol is converted into bile acids, but what is important is you know the regulatory enzyme, and the regulatory enzyme is 7 alpha hydroxylase, and please remember that this requires cytochrome P450 mixed function oxidase. There is a side chain cleavage followed by conjugation with glycine or taurine. Now secondary bile acids, where are they formed? Which are the secondary bile acids? Secondary bile acids are litho and deoxycholic acids. Remember, litho and deoxycholic acids are the secondary bile acids. Where are they formed? They are usually formed by intestinal bacterial modification. Again, first the primary bile salts are deconjugated, then they are undergo dehydroxylation to result in the formation of deoxy or lithocholic acid. Now, there is one small thing about these. When they are released, they undergo enterohepatic circulation. What does it mean? From the intestine, they go to the liver. So 98% of bile acids are recycled. 15 to 30 grams of bile salts are secreted into intestine. 0.5 is lost in feces. Rest is absorbed through the portal vein. Now there is one important drug, that is cholestyramine. This is a bile acid binding resin and prevents enterohepatic circulation. Hence, when cholestyramine is given, more cholesterol is converted to bile acids and thereby it decreases the cholesterol levels. In the next session, I'll talk to you about hypercholesterolemia. Thank you.